In this video, I will cover the rules and regulations for working in a classified laboratory. What is a genetically modified organism? These are organisms that we have altered through molecular techniques and through the process of transformation. We introduce a fragment of foreign DNA or recombinant DNA that the microorganism takes up. Basically, we are talking about a microorganism that harbors new genes or alterations to genes that do not occur in this form in nature. We use these strains that we create for testing our scientific hypotheses in the laboratory. We consider them a biological agent with an associated potential risk. It is impossible to predict how these strains will impact the environment, and so we must contain them in a closed working environment to prevent release of these microorganisms. We do that with a set of precautions that I'll cover in this lecture. When we are done working with GMOs, we must consider either storing them for future studies or disposing of them according to the guidelines. The settings where we apply biological agents is, for example, cultivation. Here, microorganisms will proliferate in a container together with a lot of substrate, and we usually end up with many more cells than when we inoculated the culture. And the microorganisms may not behave the same as if we encounter them in nature. We have two types of cultivation that we consider: liquid cultivation in broth, and then we have solid cultivation on agar plates. Are all GMOs dangerous? No. We classify them according to risk assessment guidelines. There are 4 classifications. Class 1 comprises microorganisms that are not suspected to be harmful to humans. Some examples are Escherichia coli, Saccharomyces cerevisiae, some fungi, etc. Before we begin working on a project with GMOs, your local project leaders and researchers must notify the proper authorities. There are 3 more classes. In class 2, we can work with agents that can infect humans but rarely cause disease. In class 3, we can work with known pathogens that can cause serious harm or illness. And class 4 is reserved for only the most hazardous agents such as Ebola virus. The precautions we must apply increase with biosafety level. And since you are likely to work only in a laboratory with the classification of biosafety level 1, there is little risk. And so we do not need to consider the same precautions as the higher level classifications do. What I cover in this video are the rules and regulations that we must abide to when we work in the molecular lab. The content is covered in the Health, Safety and Environment Manual, and I recommend you download this document and read it before going into the laboratory. As the molecular lab is a classified area, we only allow people to enter if they have a proper work permit. And if you're a student, then you must pass the Health, Safety and Environment exam from the university each semester. That also means that visitors, if there are any, must be accompanied by staff members. We provide lab coats, and they must always be worn on the inside of the lab area and kept buttoned at all times. This also applies to any visitors. The use of mobile devices and tablets is prohibited. Keep it inside your pocket on the inside of the lab coat. Personal computers must not be taken in and out of the lab. We have a dedicated computer present in the laboratory. I recommend bringing a physical lab book for keeping notes. It goes without saying that we do not permit eating, drinking, or smoking. We also discourage the use of lip balm, chewing gum, etc. The labs are to be kept clean and tidy. And that means you can only leave equipment which is relevant to the work permitted in the laboratory. If you have personal samples for your project, then you can use the designated cupboards, but not the workplace tables. The tables and lab benches are cleaned by those who use them, and that means you. Some good practices are to disinfect your workstation every day when you leave and wipe down the table and surfaces with disinfectant. If you use paper towels, they are to be considered as GMO waste and must be disposed as such. Remember to wash your hands or disinfect them when they have come in contact with biological agents or GMO material, or if a spill has occurred. Always wash your hands or disinfect them before leaving the laboratory. Doors should be kept closed at all times for ventilation effectiveness. Irrelevant materials are not permitted in the labs, and this includes technical devices not used for work, your own bags or clothing. All plain cardboard boxes should be replaced by plastic boxes. Materials which are difficult to clean can be wrapped in plastic. Mouth pipetting is not permitted. Note that in Esbjerg we have a special set of lab coats marked Svampelab, which are for the classified lab area only. Do not remove them from the classified area. They are washed by the staff, so you don't need to bring them home. Disposable gloves are provided in all labs and are to be used when there is a risk of biologically active material and when using reagents that pose a health risk. An example would be ethidium bromide, antibiotics, etc. The used gloves are treated as Class 1 GMO waste and should be disposed as such. Goggles are rarely necessary in the laboratory, but they are available. Make sure you follow the correct regulations regarding safety when handling chemicals. In the molecular lab, you will encounter something called a Laminate airflow bench. They are equipped with a ventilation system from an external source. The cabinet creates a sterile working environment for your cell cultures and experiments. Please note that the airflow goes from the top of the bench and out into the area where you are sitting, so the lab bench does not protect you, but it protects your sample from cross-contamination. I just want to cement that a lab bench is not the same as a fume hood. Lab benches cannot be used when working with volatile hazardous chemicals. You must, in that case, use a fume hood. The fume hood protects you from volatiles. A lab bench protects the sample and not you. If you wish to store or transport the material from the classified area, you must do so in closed containers. Bags or boxes are to be labeled with their contents, name and date. Freezers belonging to the classification but standing outside the area are also marked with GMO Class 1. Before transporting material, your material must be packed in a typically closed, unbreakable container such as these on the slide. Before work starts, instruction must be given on the use of disinfectants, emergency showers, eye wash stations, first aid equipment, along with the evacuation plans and escape routes. Such instructions are arranged by your local health and safety responsible.